Hello everyone, and welcome back to this online lecture series. This video will focus on enzymes and their role in metabolism. We will cover how enzymes act as catalysts, including how they affect activation energy, substrate specificity of enzymes, things that affect reactivity such as temperature and pH, cofactors and coenzymes, enzyme inhibitors, and enzyme regulation. After this video, you will be able to explain the role of enzymes in cellular metabolism. Particularly, you'll define the structure and function of enzymes in biological systems, explain the general models of enzyme activity, describe the function of enzyme inhibitors and how allosteric regulators affect enzyme activity, and explain how local conditions affect enzymes. So, let's begin with what an enzyme is. We know that enzymes are absolutely crucial to our metabolism. In this metabolic map, every reaction is catalyzed by an enzyme, and every enzyme is specific to its reaction. It's estimated that 75,000 enzymes exist in the human body. Their purpose is to speed up these chemical reactions. They enable us to see, hear, feel, move, and think. Every organ, every tissue, and all 100 trillion cells in our body depend upon the reaction of metabolic enzymes. Without these metabolic enzymes, cellular life would cease to exist. Enzymes are defined as catalytic proteins that speed up metabolic reactions. We're going to break this definition down. First, recall that proteins are made of amino acids and need to be folded properly for them to function. This image of a protein shows the 3D structure. We can see the alpha helices and the beta-pleated sheets, shown as arrows. Most enzymes are made of proteins. A catalyst is a chemical agent that speeds up a reaction without being consumed by that reaction. So a catalyst will perform its function, then re-emerge unused, unharmed, unaltered, and can therefore be used again. An enzyme is a catalytic protein. It performs its function over and over again without being consumed. In fact, enzymes can catalyze reactions up to 1 million per second. An example of an enzyme is sucrase, which catalyzes the hydrolysis of the sugar sucrose. Sucrose is a disaccharide comprised of glucose and fructose. Remember that sugars and carbohydrates end in ose, O-S-E, while enzymes end in ase, A-S-E. Oftentimes, enzymes are named after the reaction they catalyze, as we see here. Taking this one step further, how do enzymes speed up metabolic reactions? Answer is by lowering the energy barriers for the reaction. Here is a reaction coordinate for this reaction. If you need a refresher on exergonic reactions, see part 1 of this lecture. Here, sucrose begins with a high free energy, G. The products have a lower free energy, so this is an exergonic reaction. It releases energy and is spontaneous. This means this reaction will happen on its own over time, even without an enzyme. However, it will happen way too slowly, which is why we need enzymes. In this sucrase reaction, the black line represents how the energy of the reaction changes over time. Notice that bump in energy. It's called the activation energy. And in order for the reaction to proceed, it needs to absorb a little bit of energy from its surroundings. Usually the heat of the surrounding environment is enough. Then the reaction can proceed. Now look at the red line. This is how the reaction proceeds with the use of an enzyme. So what happened here? The activation energy was decreased. Less energy is needed before the reaction occurs, which helps speed it up. Here are general reaction coordinates for an exergonic reaction and an endergonic reaction with and without enzyme catalysts. The exergonic reaction on the left looks just like the sucrase reaction we just saw. The delta G here will be negative, and so it is spontaneous and exergonic. The enzyme catalyst lowers the activation energy, which will speed up this reaction. The same is shown on the right with the endergonic reaction. This reaction requires energy input, so it will have a positive delta G and is endergonic. This reaction will be coupled to a separate exergonic reaction, most likely the hydrolysis of ATP to proceed. If you need a refresher on that, see part 1 of this lecture as well. The presence of an enzyme catalyst in both of these situations lowers the activation energy and it will proceed faster. So what is activation energy? Remember, every chemical reaction between molecules involves the bond breaking and bond forming. And there's an initial energy that's needed to start a chemical reaction, and this is called the free energy of activation or the activation energy, and it's abbreviated with a capital E and a subscript capital A. Activation energy is often supplied in the form of thermal energy, which the reactant molecules can absorb from their surroundings. Recall from part 1 of this lecture that thermal energy is a form of kinetic energy, and it's associated with the random movement of atoms or molecules. Heat is a type of thermal energy which is transferred between objects, which is why you can feel heat when you are near another living organism. In this clip, we see the red molecules possess more thermal energy. They're moving much faster and are passing it on to the blue molecules. So now we know that enzymes speed up reactions by lowering the activation energy. But why is an activation energy even required? Well, to get reactants into a state that allows those bonds to break, the molecule must be somewhat contorted. Small energy input is required to achieve this contorted state, and this contorted state is called the transition state, and it is a high energy, unstable state. Reactants don't last very long in their transition state, and they'll proceed quickly to the reaction's next steps. In this example, the reactants must have contained high energy in their bonds, so there is a release of energy, and the free energy falls down to the products, which have lower free energy, G. The transition state of the reaction always exists at a higher energy state than the reactants. So the activation energy is always positive. There is always, whether we're talking about an endergonic reaction or an exergonic reaction, there is always a positive activation energy, a little bit of energy that's needed for those molecules to absorb in order to get them to that transition state. Note that enzymes do not affect the change in free energy delta G. Write that down. They don't affect the change in free energy delta G. Remember, delta G is only affected by enthalpy, temperature, and entropy. Do you see anything in this formula about enzymes? Enzymes don't affect delta G. Instead, enzymes hasten the reactions that would occur eventually anyway. Let's get into some enzyme terms. I mentioned while showing you the metabolic map that enzymes are highly specific in what they can bind and what reaction they can perform. The reactant that an enzyme acts on is called the enzyme's substrate. When the enzyme binds to its substrate, it forms an enzyme-substrate complex. And while bound, the activity of the enzyme converts the substrate to its product. The active site is the region on the enzyme where the substrate binds. This is an example of an induced fit model, which we'll look at in a bit. To reiterate, reactions catalyzed by each enzyme are very specific. To break down the sugar lactose, our bodies use the enzyme lactase. To break down the sugar sucrose, what enzymes do our bodies use? You should recall that the enzyme used here is sucrase. To understand how an enzyme works, we have to focus on the active site, because this is where catalysis occurs. In an enzymatic reaction, a substrate binds to the active site of the enzyme. There are a few theories for how this happens. The first is the induced fit model. In this theory, the initial interaction between enzyme and substrate is relatively weak, but these weak interactions will rapidly induce a conformational change in the enzyme, which will strengthen binding. This simply means that the enzyme is induced to change its shape to better fit the substrate. To contrast this, another theory is the lock and key model, and this is similar to how a key has to be the correct one to fit a lock. No reaction takes place if an incorrect substrate tries to bind. This process is very specified in that only a particular key can fit into the keyhole of the lock. If the key is any smaller, larger, or simply a different shape, then it does not fit the keyhole and the reaction cannot take place. Generally, it's believed that enzyme reactions are a mix of both induced fit and lock and key. So let's say our substrate has bound to our enzyme, creating the enzyme substrate complex. Then what? How does the active site lower the activation energy for catalysis? Well, there are 4 ways that an enzyme can lower the activation energy barrier: orienting substrates correctly, straining substrate bonds, providing a favorable microenvironment, and covalently bonding to the substrate. The active site provides a template on which the substrate molecules can come together in the proper orientation for a reaction to occur. We see here the binding site and a catalytic site. The enzyme helps to orient the substrate properly for catalysis. The enzyme is also able to grab on to that bound substrate and may physically stretch them towards their high energy transition state form. And this will stress and bend the bonds which need to be broken during the reaction. This reduces the amount of free energy that must be absorbed to achieve that transition state. The active site can also provide a favorable microenvironment that is more conducive to the desired enzymatic reaction. For example, if the active site has amino acids that are acidic R groups, the active site may be a pocket which holds low pH, so it would be more acidic, so that that amino acid is in a better microenvironment. And finally, enzymes may covalently bond to the substrate to aid the enzymatic reaction. This is a big deal because covalent bonds are stronger than the weaker interactions that are used to accomplish the previous 3 tasks. Let's review catalysts before we continue. Enzymes are catalytic proteins that speed up reactions which would already occur. They function by lowering the activation energy. However, they do not affect change in energy delta G. Enzymes are not consumed by the reaction and are used repeatedly in catalytic cycles. Now let's summarize the specifics of enzymes. A substance that helps a chemical reaction to occur is a catalyst, and the special molecules that catalyze biochemical reactions are enzymes. Almost all enzymes are proteins comprised of amino acids, and they perform the critical task of lowering the activation energies of chemical reactions inside of the cell. Enzymes do this by binding to the reactant molecules and holding them in such a way as to make the chemical bond breaking and bond forming process take place more rapidly. Enzymes do not change the reaction's delta G. In other words, they do not change whether a reaction is exergonic and spontaneous or endergonic. This is because they do not change the reactants or product's free energy. They only reduce the activation energy required to reach the transition state. As we know, the purpose of an enzyme is to speed up the rate of the reaction, and there are ways that we can manipulate the rate of the enzyme's reaction. Of course, the two major components of a reaction are the enzyme and the substrate. The rate of the enzyme catalyzed reaction can be sped up by increasing substrate concentration. Here is a graph of enzyme activity versus substrate concentration. Notice that as we increase the concentration of a substrate by adding more, the enzyme activity also increases. When all enzyme molecules have their active sites engaged, the enzyme is then said to be saturated. If the enzyme is saturated, the reaction rate can only be sped up by adding more enzyme. Enzymes are extremely fast-acting, and some are able to even react at 1,000,000 times per second, and they emerge from their reactions in their original form. Very small amounts of enzyme can have huge metabolic effects because they're used repeatedly in those catalytic cycles. Now, we can also change how efficiently the enzyme functions. An enzyme's activity can be affected by a few things. They can be affected by general environment factors such as temperature and pH, and also chemicals that specifically influence the enzyme. Recall from the lecture on macromolecules that the 3D structure of proteins are very sensitive to their environments and they prefer optimal conditions to function. When proteins are not in optimal conditions, they may unfold as seen here, and this process is called denaturation. Optimal conditions favor the most active shape for the enzyme molecule. Each enzyme has an optimal temperature in which it can function. For our enzymes, the optimal temperature is body temperature, which is around 37 degrees Celsius. For a thermophile, which is a heat-loving bacteria, the optimal temperature will be much higher. It's around 75 degrees Celsius. From this graph, we see that increasing temperature increases the rate of reaction up until the optimal temperature, and this is partly because the substrates collide with the active sites more frequently because the molecules are moving more rapidly. Higher temperature means higher thermal energy, and thermal energy is kinetic energy. So there's just more movement here. After optimal temperature is reached, the rate of reaction drops sharply as the temperature is raised. And by the way, this is also why spiking a temperature can be bad for us. It damages our enzymes as our internal body temperature increases. Each enzyme also has an optimal pH in which it can function. Pepsin is a stomach enzyme that thrives in an acidic, low pH environment. Activity is optimal around pH 2. The rate of reaction drops as you increase above pH 2 or decrease below pH 2. So as we move away from that optimal pH, the protein is more and more likely to denature, which will change its shape and therefore its function. Chemicals can also influence the enzyme activity. Antibiotics are chemicals that inhibit enzyme activity. For example, penicillin irreversibly inhibits an enzyme called transpeptidase, which is important for synthesizing bacterial cell walls. Without active transpeptidase, bacteria can't divide. So as we've just seen, pH, temperature, and substrate concentration are all factors that can affect enzyme reaction. Enzymes may also rely on helpers to perform their functions. Many enzymes don't work optimally or even at all unless bound to other specific non-protein helper molecules, either temporarily through ionic or hydrogen bonding or permanently through stronger covalent bonds. Cofactors are non-protein enzyme helpers. Cofactors can either be organic or non-organic. Remember, organic simply means the molecule contains carbon. Inorganic factors could be metals in ionic form, such as magnesium, copper, and manganese. Notice they all have a charge, making them ions. Iron-sulfur clusters are a major component of redox reactions of the electron transport chain in mitochondria and chloroplast, and we'll learn about redox reactions in the electron transport chain in the next lecture. An organic cofactor is also called a coenzyme. Coenzymes include vitamins and forms of vitamins. We'll learn about one in the next lecture called coenzyme A. As we know, the body is a highly regulated organic machine. Cellular needs and conditions vary from cell to cell, and they even change within individual cells over time. For example, a digestive cell in your gut works much harder to process and break down nutrients right after you've had a meal compared to hours before eating a meal. As cellular demands and conditions vary, so do the amounts and functionality of different enzymes. If a cell's metabolic pathways were not tightly regulated, we would have chemical chaos in the body. The body has two main mechanisms to regulate the amounts and functionality of enzymes. A cell can do this by switching on or off the genes that encode specific enzymes, or by regulating the activity of enzymes that are already made. These ultimately determine the rates at which biochemical reactions occur. Notice that altering gene expression is the slow way. Can you figure out why this is? We've touched on gene expression very superficially, and we'll learn it in depth in a later lecture, but for now, recall that gene expression is DNA to RNA to protein. It takes time and energy to do this, and it's a highly complex biological process. Altering enzyme activity of enzymes that are already made is a more rapid form of enzyme regulation. These are things that we do to the actual enzyme that affect its activity. We can make covalent modifications to turn the enzyme on or off, and we can utilize allosteric regulation to control enzyme kinetics. We're going to focus on rapid control activity and allosteric regulation for the rest of this lecture. We'll start with a basic model of enzyme inhibition. Enzymes can be regulated in ways that either promote or reduce their activity. Here we see two mechanisms to inhibit enzyme activity. Image A shows a normal enzyme binding to its substrate. Image B shows competitive inhibition. Competitive inhibitors bind to the active site of an enzyme, competing with the substrate and ultimately blocking the substrate from binding and therefore inhibiting the reaction. It's called competitive because the two molecules are competing for access to the same active site. Image C shows non-competitive inhibition. Non-competitive inhibitors bind to another part of the enzyme other than the active site, causing the enzyme to change shape and making the active site less effective. Here, the orange inhibitor is binding at the opposite side of the enzyme, and we see that the active site has changed shape. Now, the substrate can't bind to the active site, and this reaction is inhibited. Examples of inhibitors are toxins, poisons, pesticides, and antibiotics. It's good to be able to read and analyze various graphs in science courses. Here we see another type of graph plotting reaction rate versus substrate concentration. It's comparing the rates of reaction for enzymes with just substrate present, or with competitive inhibitor, or with a non-competitive inhibitor present. We already know that substrate concentration can affect the rate of reaction up until the point at which the enzyme becomes saturated. But why do reactions in the presence of competitive and non-competitive inhibitors have different rates? And why does competitive look different from non-competitive? Let's consider competitive first. A competitive inhibitor is competing for access to the active site, and it's competing with the substrate for this access. So it makes sense that if we increase the amount of substrate, it'll have a higher chance of being the molecule that binds to the active site. But this only increases the rate up to a certain substrate concentration. Now contrast this to the non-competitive inhibitor. If the non-competitive inhibitor is bound, the active site changes shape, inhibiting binding of the substrate. It doesn't matter how much substrate is present, it won't change the fact that the active site is not the correct shape. Therefore, adding more substrate will not increase the rate of reaction. The reaction rate peaks much lower than for the other two scenarios, even if we continue to add substrate. The non-competitive inhibitor that we just learned about is an example of allosteric inhibition. Allosteric regulation occurs when a regulatory molecule binds to a protein or an enzyme at one side and affects the enzyme's function at another. The word allo means other. Allosteric regulation may either inhibit or stimulate an enzyme's activity. In this image, we see an example of both allosteric inhibition and allosteric activation. Both involve a molecule binding to a site other than the active site. The allosteric inhibitor caused the active site to change into a shape that could not bind the substrate. This is non-competitive inhibition. The allosteric activator did the opposite. When it binds to the allosteric site, the active site is altered in a favorable way to allow the substrate to bind. Most allosterically regulated enzymes are made from polypeptide subunits, each with its own active site. The enzyme complex has active and inactive forms. The binding of an activator stabilizes the active form of that enzyme. We see this with the green activator molecule here. The binding of an inhibitor stabilizes the inactive form of the enzyme. We see this with the red inhibitor molecule here. Substrates are now unable to bind when the enzyme is in its inactive form. The enzymes that our body needs to function are susceptible to inhibition. Here are a few examples, real-life examples for you. Heavy metals are poisonous, often because of their ability to non-competitively inhibit our enzymes. Mercury, copper, lead, and silver can all react with a particular amino acid that's present in our proteins, and by doing so, they irreversibly inhibit enzyme activity. Natural products like glycoalkaloids from nightshade plants can exhibit toxic effects through their ability to inhibit enzymes like acetylcholinesterase, which is an enzyme required for muscle movement. Pesticides like organophosphates and carbamates inhibit acetylcholinesterase and are toxic to all animals, humans, and insects. Now, you probably don't need to know these details for your class, but it's always good to have real-life examples. Cooperativity is another form of allosteric regulation, and this can amplify enzyme activity. In cooperativity, one substrate molecule primes an enzyme to act on additional substrate molecules more readily. In this image, one blue substrate molecule binds to the inactive form of the enzyme, and this causes the enzyme to change form to the active form, and the active form is now stabilized. Now, other substrate molecules can easily bind to this enzyme. Again, this is a form of amplifying enzyme activity. Cooperativity is allosteric because binding by a substrate to one active site affects the catalysis in a different active site. Of course, the way our body is able to monitor and control enzyme activity is via feedback regulation. We're always going to return to this concept because it's literally the mechanism behind everything in the body. In feedback inhibition, the end product of a metabolic pathway shuts down the pathway. In this image, green means go, red means stop. An original input activates the reaction of A, which results in B being created. B is the end of this metabolic pathway, and it is acting as an inhibitor to shut down the pathway. Feedback inhibition prevents a cell from wasting chemical resources by synthesizing too much of a product, more than is necessary. Here's a more detailed example of feedback inhibition. Here, enzyme 1 binds the substrate, which creates intermediate A. Intermediate A acts as a substrate on enzyme 2, which creates intermediate B. Intermediate B acts as a substrate on enzyme 3, which produces our end product, the yellow square. The yellow square is the end of this metabolic pathway and will act as an allosteric inhibitor on enzyme 1, effectively shutting down this metabolic pathway. Here's a real-life example to drive the concept home. We have an enzyme called threonine deaminase. Threonine deaminase binds to its substrate threonine to create intermediate A. Intermediate A is part of this metabolic pathway shown here, which eventually creates the end product isoleucine, which is an amino acid. Once our body has created enough end product, isoleucine, it will bind to an allosteric site on the enzyme threonine deaminase to non-competitively inhibit the reaction. Now here's the important part. Once the body has used up all of the end product, there will be no more isoleucine to inhibit the enzyme, and the metabolic pathway will be reactivated again to create more isoleucine. So this is a feedback regulation cycle. We often find enzymes for particular metabolic pathways localized together within the cell. Structures within the cell help bring order to metabolic pathways. For example, some enzymes act as structural components of membranes. In eukaryotic cells, some enzymes reside in specific organelles. For example, enzymes for cellular respiration are located in mitochondria, as seen here in this image of the mitochondrial electron transport chain. Again, we're going to see this in the next lecture. To wrap up this lecture, let's discuss the evolution of enzymes. Enzymes are proteins whose structure are encoded by genes in the DNA. Changes to the DNA, which we call mutations, lead to changes in the amino acid composition of our protein. If we have altered amino acids, particularly if they're at the active site, this can result in new enzyme activity or altered substrate specificity. Now, if this mutation is beneficial, natural selection will favor it and it will be passed down, and we will keep this mutation. This is the basic mechanism of natural selection. beneficial random mutations are naturally selected and passed on to new generations. And that wraps up this lecture and this chapter. Check out this mind map of enzymes and regulation, and I'll see you next lecture where we will learn cellular respiration, glycolysis, the citric acid cycle, oxidative phosphorylation, and fermentation.